Namaste. So very important thing for any bioinformatician or any researcher, data is very, very important. If you do wet lab based analysis, we generate the data in the lab and we do the analysis and we get the results and we do the discussion. But when you go to any sort of bioinformatic research or computer aided drug designing, all this research, we try to get the data from the various databases. Okay, we try, there are databases available where information is stored and we retrieve the data and we'll start working on that. Maybe all of us in this pandemic situation, people, you could see, go to Google Scholar and search for molecular docking, SARS-CoV-2 or COVID-19. You can find thousands of papers. Most of them are bioinformatics based research. As Dr. Tirmal was telling, people are trying to repurpose the drugs rather than finding new drugs. When you're thinking of repurposing the drug, from where, what source you are going to get the information? That's what we call it as primary source, secondary source, tertiary sources, and all. Maybe you have seen a paper published in Nature journal. You directly read that paper, it becomes a primary source because the authors or researchers have directly published the data in reputed journal, and you read that data, it becomes a primary source. Sometimes you could see some newspapers and all will publish this particular lab has developed vaccine for COVID-19. So, and at the end, they give you a reference, it is published in particular journal. So then it becomes a secondary sources, means it is like a derived source. They have taken from other source. So what I would like to say is, any information you collect, it should be an authenticated data. You cannot randomly choose the data because we know today the databases, a lot of databases are created. Maybe some are authenticated, some are not authenticated. So today we discuss about few topics. Maybe for few participants, maybe it is redundant. They might be aware of this kind of databases, but most of the participants are students. We thought like we give some glimpse of these databases. I will not go through into what is computer-aided drug designing and all because so many of the speakers have discussed about what is this computer-aided drug discovery. And this is the workflow of computer-aided drug discovery. When the moment you talk computer-aided drug discovery, the data is very much important. So it might be structure-based design, it may be computer-based drug design. Everywhere you need a data. Structure-based drug design, you need a protein structure. And once you have a protein structure, again, problems are many because if you go to UniProt database, it is a sequence database, protein sequence database. There are millions of protein sequences available in UniProt database, but there are very few protein structure available. Few in the sense that if you go to PDB database, there are few structures are available. Why the structure data is less? Because solving structures are always bit difficult. There are people going to solve structures using X-ray crystallography. There's a well-known methodology. And next one is NMR. And next one is electrocryomicroscopy. Okay, but always people try to take X-ray structures because the quality of the structure is good, and the protein you make it static, and you're going to resolve the particular positions of atoms and all. NMR and all, what happens? It happens in a solution. Sometimes what the protein in a dynamic state, you cannot find out an exact structures. That problem might be there, but many a times some structures cannot be crystallized. That time definitely people go for NMR or electron cryomicroscopy. And even COVID also, we could see some structures are solved using X-ray, NMR, and even electrocryomicroscopy. So that is what sometimes what happens if there are no structures available, people go for homology modeling. Okay, people do homology modeling. There are tools available. Modeler tool is available. Swiss model is available. They use such kind of tools and they try to predict the structure based on homology. Means similar structures are available in the database. They use such databases and they try to predict the structure of databases. And there are again databases for this homology modeling. There are Modbase is there, Swiss model is there. Means there are databases. People try to predict the structures and upload. You no need to again do the prediction. People do the prediction and upload some places. So you can download, and because when you are new, you are not aware how to do homology modeling. Definitely you can take from those particular sources and you can use it. That is one about structure based data. And yes, once you have a protein, if a structure is available in the database protein, what happens? Sometimes with a co-crystallized ligand, they will solve the structures. That time, finding an active site of the protein is not difficult. You can easily find out where are the ligands are bound. That can be taken as an active site prediction. But when you predict a structure, how do you take up the active site data? So what you can do is you try to predict the binding site again. So you need, there are again databases.
Sir, TPA, sir.
Sir.
Make it louder, sir, because audio is not clear, sir.
Hello. Now it is audible, sir?
Now it is audible, sir. Before very low, sir, audio.
Sure. I'll take care. Thank you. Okay. So now the binding site prediction. So next what happens? You need to predict the binding site. Okay. There are again databases are available which will help us to get the binding data also based on homology and all. Next, when it comes to ligand based drug design, again there are a lot of ligand based means definitely you should have a ligand molecules, as some speakers are talking about drug repurposing and all. So from where do you get the drug data? Where it is available? So whether the drug is approved or simply just identified by someone else, because phytochemical compounds, people are these days talking about lot about phytochemical, because every country or every religion or everything, they have their own tradition, science, and they have their own medicine they use. You have India, Ayurvedic medicine is there. You go to China, they have traditional Chinese medicine is there. Like that, you go to any other Western country, Eastern countries, they have their own medicine. The traditional medicines are there. Means that plant-based medicine. So okay, once you have a plant-based medicine, because difficulty in plant-based medicine is you get a holistic approach. And it is an holistic approach. Like you take a whole plant as a medicine, means you are extracting, taking, because otherwise the other method is reductionist approach. Means we try to isolate a compound which is having an activity. Okay, the two ways. One is the reductionist approach, another one is a or like you take a holistic approach. So then people will identify this many plant compounds are available in the plants. Okay, they do the LCMS or any analytical technique they might employ, and I try to identify the compounds present in the particular plant. And what about the drug repurposing I was talking? You want what are the approved drugs? Like for example, FDA approved drugs. Where do I get in information? There are databases which will help us to retrieve the data. Yes, sometimes what is the difficulty? You will identify plant-based material. You take that one particular plant. So you take Tinophora cardifolia. Hello.
Audio gone, sir. Audio gone, sir.
Hello, sir. Now.
Now it's hearable, sir. Audible, sir.
Fine. So now, so whenever you talk about, for example, Tinophora cardifolia, so they identify which compound will give you the activity. So they identify the compound. You do LCMS, all these things. You will analysis. You will perform. You identify one particular compound. Fine. Now the problem is you identify one particular compound. Now when you go for wet lab analysis, like or else you go for in vitro, this is in silico. Whatever you perform in computer is an in silico. Next level you are going to move to the in vitro or in vivo studies. You need to have that particular compound. So now you have to start purifying that particular compound or synthesizing that particular compound. It might be tedious. Then there are databases available which will help you to buy the compounds. Commercial databases are available. That's what, whenever you're working on drug design, if it is available commercially, it is better, easy to carry out further research. Why many times the computational drug discovery fails is everything goes to publication. That's it. It will not take to the next laboratory level. Why these are the difficulties? Because computer, anything can be done. Whatever you imagine, you can perform an analysis or mutations you can create a compound. Anything can be done in a computers, but you need to take up to the next level and prove whether it is a drug is working or not. That time, if the protein is not available, even proteins are required, right? You, for example, you target some SARS-CoV, you take, you wanted to work, you need to perform some enzyme assay. You need that particular protein, you take in a STO, you take in inhibitors, you do the enzyme kinetics, you identify the binding affinity, everything you identify. But if you don't have the protein structure itself, or if you don't have ligand information itself, so how do you perform the further analysis? How do you perform the further analysis? So we need to take into consideration of all these things. That's what whenever you do bioinformatics research or computer-aided drug discovery, we need to understand first these informations are available. Your aim is to just make one research paper, publish in journal. Maybe you will not bother about all such things, but you are serious enough to identify some potential drug for that disease. So these kind of things should be keep it in mind. Next one is any bioinformatician, it is now interconnected. It's not like medicinal chemistry or bioinformatics, genomics, proteomics, wide range of today biological science is so advanced, so it is not difficult for us to analyze the things. Earlier, how the disease is like happening, we don't know. The pathology we don't know, but today we have understood the molecular biology. We have well established molecular biology, and whatever the sir was telling about mutations and all, we have NGS, next generation sequencing technology is there. Whole proteome of that organism can be known by the proteomic analysis. Today, the things have become cheaper also and easily available, so you can perform. This is the one meta database I can see, Swiss Institute of Bioinformatics, that is XPACY we call. So this XPC Bioinformatics Resource Portal, those who are interested, they can browse this XPC server. So you can see n number of literature like databases are available. I will not go in detail about each one because it is not required also, because whichever area you're interested, for example, you're interested in proteomics, genomics, structural analysis, systems biology, whichever is area of your interest, you can go to that and you can find out. They have listed out the complete information, details, what are the softwares available, what are the databases available. You just go there and you can find out the information. And next one is computational map of conceptual map of computational drug discovery. So yes, people will explain later in next coming talks, like this is the flow of computational drug discovery or any drug discovery process. Target identification. Question comes to is, where do I find the target? What is my target? Which one should I use it as a target? Sometimes we go with literature survey, we perform, this might be a potential target. COVID-19 also, we have understood there are a lot of structural proteins are there, non-structural proteins are there. Which one to target? Whether is it feasible to target structural proteins, or is it feasible to target non-structural proteins? So how do you do it? Because non-structural proteins, basically they are kind of enzymes and all. Structural proteins are like membrane proteins, receptors and all. So which one you are going to choose? Where this information is available, target information is available. So next one, next one, hit identification. Yes, once you have a target, next we want a ligand molecules. As I was telling about, you need to next level, you go for finding ligands. So I will not go into detail about all these things, but each, why I am showing these pictures, because each step you need a data. So some data is required. That for data, that data, where do you identify? Those who are interested to work on drugs like drug molecule, this is one more best source for those who are working in medicinal chemistry or computational biology or computational drug design. Click to drug is also again a part of Swiss Institute of Bioinformatics. Here you can see databases, chemical structure representation, molecular modeling, homology modeling. So means each category, what are the databases you require? As I told, when you want to start doing a computer-aided drug discovery, you need to have first target information. Next one is ligand information. Next one is sometimes when you go for QSAR analysis, you need to have binding affinities of that ligand with that particular protein. And next level, protein ligand interaction, protein interaction. So it is an integrated network. Many information are required when you do a drug design. It is not to isolate some drug, take some COVID enzyme, for example, main protease you have done some activity analysis, but in silico level, everything can be fine. Even the dynamics, molecular dynamics, what we run works very well when you do it in a computer, but real dynamics when it is in a in vitro or in vivo, definitely it changes. So you need to understand that interaction. You are targeting one particular target, especially when it comes to genetic diseases or some diseases. You try to give some drugs to it. Even you take headache, a tablet for headache also, it is having some effect on other proteins also, because interrelated. The protein interaction is there, networking is there. If you target something, it might affect other one. That's what target identification is very, very crucial. And I hope all of you are aware about antibiotics. Okay, how do you choose an antibiotic? What target you choose? Penicillin you take, the target is like cell wall synthesis enzyme it targets, because if we don't have cell wall, so definitely you can target that. If we have the same enzyme, definitely you cannot do it. Many times antibacterial, whenever you go for antibacterial, the bacteria, why they sustain in harsh environment also, because they have a capacity to synthesize their micronutrients. Most of the micronutrients we depend upon some sources for micronutrients, especially vitamins or some amino acids and all. But bacteria or any pathogens, usually the beauty of those biological system is they have a capacity to synthesize some of these micronutrients. So what people try to do, they try to target these kind of things. If you target those particular pathways, definitely you can find out a efficient drug. So that is what target identification is. Not only from the database, you should have a thorough grounding or thorough literature survey, then you will understand. So next one, this series, you please go through this. You just go to www.clicktodrug.org. It will fetch you the information. Next, this is one more information and one more website, a metadatabase called vls3d.com. So those who are computational biologists, definitely it will have wonderful resource, a database, and all the information related to computational biology, not only computational biology, even bioinformatics. People who are not interested in drug design, other aspect of bioinformatics also, this is the source. A metadatabase will give you a lot of information. And yes, as I told, if I go in detail about each and every tool, it will be boring, because those who are interested in what aspect you are interested, please go to that particular website and you can fetch the data. Few databases I'll explain. Those are like new to this drug design. Chemical databases, as I told, zinc database is always best, because as I told, commercially available. The ligands, if you take from zinc database, you retrieve the data of ligands from zinc database, you perform the molecular dynamics analysis, those ligand information, that ligand you can purchase. If you purchase, the next what happens, in silico you have done an analysis. If you want to carry out in vitro analysis or in vivo analysis, this ligand molecules, you can purchase from those databases like zinc database and all. You can do the analysis. That way you can prove not only in silico you say that drug is potential, but you can also say that in vitro and in vivo analysis also be carried out, and this drug can be used for analysis. This is what a zinc database and zinc 15 database. And GMBL, that is CHEMBL, GB we call that is CHEMBL or EBI, that is EMBL, European Molecular Biology Laboratory. There also curated databases are there. This database is again might not have commercially available drug molecules. Maybe there's an information available, a database. How the Google Scholar, it will only show you the research articles. But again, if you want to read that article, you have to go to the original publisher. Okay, so you have to go that publisher again, whether the publisher is open access or you need a restricted or your institute is having access, that is a different issue. But Google Scholar will give you only the information like that. These databases also will give you the drug information, and you can download the drugs. Okay, in a different formats available, you can download, you can perform the analysis, but that might not be commercially available. You have to find a route. How can I obtain this particular drug? Even the ChemSpider, even Drug Bank is a powerful database. PubChem is part of NCBI, National Center of Biotechnology Information, and even TCM is traditional Chinese medicine. As I told, each country, they have their own traditional medicine and try to make the databases. Even Ayush in India also, Ayush made a databases. What are the medicinal plants? Because huge amount, most of the, when you take an example of artemisinin, people are aware of anti-malarial drug. Okay, that is also derived from natural source. Artemisinin is a plant, like tree, they have derived from that particular plant, a wonderful potent drug for anti-malarial. So when you look into the literature, like your own traditional medicine, so a lot of information might be available. That is what people today, they are looking into it. Ethnobiology, ethnobotany, means they are going back to that traditional knowledge and try to identify the diseases. Earlier also these diseases were existing, but how the people were managing with these kind of diseases, what they are doing. But today, present day situation, our health, our food habits are not good. That is food is a medicine. So that is what people say. Okay, food is a medicine. Food should be the medicine actually, but it's not happening. So that is why traditional medicine having a lot of potential, and we can start exploring that one. So not only tradition is that we should not only study Indian traditional system, we have opportunity to study any traditional system. That's what people try to document. And even MQL, I was telling about drug repurposing. When you want to do a drug repurposing, approved drugs, where you get an approved drugs. This is what approved drugs is a database where you can find out this kind of FDA approved drug. Once it is approved, obviously you can repurpose it because it has undergone the preclinical trial and all. So that can be significantly minimized. It can be directly used. That's what the people are, drug repurposing is a big research now. Why again waste on identifying the new drug? Because one drug has to come to market means millions of dollars or millions of rupees that need to spend, and it will come to the market. Some drugs might fail for that particular disease that might work best for other diseases. The drug repurposing, when you want to do it, you can get FDA approved drug information also. Structural databases. Yes, when you want to have a crystal structure, even ligands also a crystal, you need to have a crystal structure. So that can be get it from structural databases, ChemBank, Ligand Expo, and Cake Drug. Cake Drug is again a wonderful interaction information is available. I was telling about IMPACT. This is a curated database of Indian medicinal plants, phytochemistry, and therapeutics. So those who are interested to work on phytoceuticals or nutraceuticals, these databases will help you to understand. So what are the phytochemicals available? You can employ and study it. So antiviral drugs, people talk about, there are a lot of antiviral potential plants are available. So different tradition have different things. Some especially I want to say about Siddha medicine, they are giving Kabasura Kudini. So this again some 8 to 10 herbs they the mixture, they take the decoction and the drink. So that's they have understood. So these plants are having antimicrobial activity. Every plant does not have all the activities, but you need to identify which plant have an activity. You can definitely study this IMPACT. And aroma DB, again medicinal plants, aromatic plants, especially those who give some smells, aroma, flavors, giving fragrance. Those are all again terpenoids, equiterpenoids, essential oils and all. Essential oils also very good. They have wonderful antibacterial activities, antifungal activities. That's what olden days they were using it in creams, lotions and all. So that's it. But today they are all synthetic. They become all synthetic. That is causing lot of problems. Okay, when you use synthetic ones. Next one is Dr. Duke's photochemical ethnobotanical databases. Here also it will give you the information about many plants which are having medicinal value. And CA CAMO, that is a database of collective molecular activities of useful plants, because plant every compound may not be active. So whatever the active constituents are there, active ingredients are there, that you can extract. And this is the one interesting research article recently I went through, that is Journal of Chemoinformatics. I'm giving reference. Maybe people can note down it. You can read. It's a wonderful paper, almost 50 pages article, review on natural products databases. Those who are interested to work exclusively on natural products, maybe you can definitely go through this and identify the databases. Those who are struggling to find information, maybe this paper will give you a lot of information. You can go to this paper and you find the information. And next one is protein ligand complexes. As I told, I will not read out everything. So you can protein data bank. It's a major, is mother of like protein structures, what you can say, because everything is available. Everything in a sense, whatever people identify till date. So definitely it's available in protein data bank for any drug researcher. This is like initiation. So you can find the structures there. Even you want to predict homology through homology modeling, definitely that similar structure should be available in drug bank. And next one is like binding database, like binding data means affinity survey. Essentially, when you go for QSAR studies and a ligand-based drug design, so you need to have an affinity data, RKI, inhibition constant value, IC50 value. So binding DB, that binding database will give you those kind of information. And even PB bind, pro pairs, NL list, the list goes like this. So as I told, the metadatabases information I give, so you can just go through those databases. Definitely a lot of information is available. Yes, next challenging task is, many people in the morning also they were asking how to choose the target. So that is left to you. So how to choose a target is like, first you have to have an idea what disease you are going to try to work. Again, okay, I found SARS-CoV-2. I'm planning to work on SARS-CoV-2. What target I should use it? If I use membrane proteins, what happens as my target? If I use proteases as target, what happens? If I use some RNA dependent polymerase, RNA polymerase, okay, what will happen? So target, when you choose, you have an information. So you need to understand biology of that particular target. People try to target ACE2 receptors. Okay, target may be in a host or target may be in a pathogen. Either way, you can choose a target in host or you can choose a target in host, like a pathogen. If, for example, if you try to take a COVID protein and find a drug, it is like you are designing a drug against particular bacteria, like a virus or a bacteria. If you take any bacterial disease, it's bacteria. But you can also work on the host, because this virus entering the human is having some route. So if you block that particular route, it will not enter the body. So you can develop host, in the host also you can develop the drugs. So either way you are you going to choose a host based targets, or are you going to use a pathogen based target? So based again biology, it is like always biology no one can understand completely. It's a completely non-linear system. That's what I say. It is not a linear system. It is a completely non-linear system. Sometimes might work, sometimes it might not work. Next one is pathway databases, is very important. So where this protein is involved. When you want to target some particular protein, especially you are in some human disease or genetic diseases you want to target, this particular, as I told, there is a relationship. We say the junk DNA, it is not a junk DNA. People say that only functional DNA is 1%, 2% remain is junk DNA. It is really not a junk DNA. If it is a junk, it would have not existed in the cell. Okay, because nature has evolved. Millions of years evolution has happened. If it is a junk, just it could have deleted. It is not a junk, it's an hidden information. Some information is available, we have to retrieve. That is what people are working now on junk DNA. They have left the functional DNA. Now they are working on junk DNA. What information is available? How do I, what is the, like through the evolution, some information is stored that can be used, okay, for further analysis. I will show you few websites as I was talking. Parshram sir, this screen is visible? Parshram sir?
Sir, I'm checking. No, sir. It will take time, I think. No, it's not visible.
Okay. 1 minute. Now it is visible, sir?
Yes.
Okay. So now I will show a few websites where the information, as I was telling you, NCBI is the best source. Any bioinformatician, it's like everything is available in this, like most of the information. When you call it as a metadatabase also, they link to other databases. You can just browse this there are hundreds of databases and tools are available. And just now, Dr. Thirumal was talking about mutations. Okay, I want to work on some particular mutation. So I want to work on particular gene or protein, understand the particular mutation. This DBSNP, so single nucleotide polymorphism, this is a database. So you give any gene name, it will retrieve the particular, people would have done NGS analysis, next generation sequence analysis, and they have identified the mutations. As I told, some drug might work better for one person, that might not work for other person. Same paracetamol you take, some might work for one person, some might not work for other person. Why? Because that is the how the drug is, like that's what we say, pharmacodynamics and pharmacokinetics. Okay, what drug does to the body and what body does to the drug, these both are very important. Okay, so you have to understand any mutation happened. So sometimes mutations are good, sometimes mutations are bad. So this is one such source where you get a mutation data. So someone is working on, for example, levodopa. It is used like some of the drugs used in neurodegenerative diseases and all. It is not efficient to some person. You have to identify what mutation is reducing the activity of levodopa. Definitely go to this SNP database, type DDC. DDC is the gene name. There is a database called as genenames.org or genecards.org. These are the websites where you get a human gene information, because everything is sequenced now. Gene information is available. If you are interested to work on any particular gene, definitely go there and you can work on these databases. Like that, NCBI has a lot of databases will help you to understand. There are tools available, databases available, you can retrieve the information. And next one, those who are working on genetic diseases, you are interested to work on some genetic diseases. These are the best source, genetics home reference. This is a web source where it will help you the genes, what particular gene causes what type of disease. As you can see, each protein they have given in, each gene they are given what kind of protein it expresses. See, it is again metadatabase. What is the normal function? Health condition. So what is the syndrome, health, genetic disease related to it? What is the chromosome location? So source to other pages means it is linking you to the other pages also, relevant information. Okay, so OMIM databases, online Mendelian inheritance in man. So it will link you, and related publications also it will link you. So next one, so it's like XSpace. It is again a meta database. I was talking in my presentation. So someone is interested in systems biology, because it is gaining a lot of popularity. People are not doing conventional biology; they are going to systems biology, metabolic engineering, systems biology, synthetic biology. So these are all the tools will help you. There are a lot of tools available related to systems biology you can study. So for example, you are interested in biophysics, structural data. Definitely it is available. Open Structure, Swiss Pram. So the tools are available. You are interested in medicinal chemistry. Yes, this is available. What are the information? Swiss ADME. You want to predict some ADME. I was talking about click to drug. It's an extensive database. If you just click to drug, definitely it will give you different tools and databases available. This is a metadose. Maybe please you can write on this www.xspacey.org. Next one is, I was talking about VLS 3D. And if you click here, you can see the huge set of information or databases or resources they have tried to give, make you easy. Someone is started the drug research, you don't know where the information is available. Definitely these 2-3 sites are like wonderful. You can have every information, mostly 100% information you can available from these databases. You can just go there, whichever you are interested, you can just click and you can work on that. And next one, that's what click to drug I was taking. If you just click on that, you can see the database I took extracted from the same resource. Again, I copy paste in my PPT. You can see this, the huge list. If you go to databases, chemical databases, you can see there are tens and hundreds of compound databases are available. Each one is having some advantages and disadvantages. So you can choose it. There is no, again, question like if you ask me what is the criteria to choose, again, it is the criteria is you. You can plan and you can choose what you want. Okay. So next one, database handling. Sometimes the download, once you download the data, you need to manage the data, right? Okay, just sometimes what happens, once you download particular data, it may not be feasible for you to work in another platform. You need to convert from one platform to another platform. Like for example, Word file use docx, ODT file, PDF. So the same software cannot, MS Office cannot open PDF. You need to have other software. So you can convert from one forum to other forum. So that database handling will help you. And protein ligand complexes I was telling you. So information about protein ligand definitely is available. Protein data bank is the best source I was telling you. And there are some other databases available. This is therapeutic target database. Those who are searching for targets, maybe therapeutic target database will help you a lot. And pathway databases, that interaction of the protein, particular ligand and all, you can take. And next one is chemical structures. As I was talking, ligand. Now we are like, okay, we got a target, now ligand. Where do I get ligand? Yes, there are a lot of databases available. As I already told, ligand information you can download from there. If you don't have the structure available in the database, someone would have identified, okay, they have in their lab, they identified a new molecule from this particular plant. Okay, they have reported in the literature. Now there is no 3D structure available. What you can do is you can draw those structures. You can draw those structures and you can convert it to whatever format you require for further analysis. You can do it. There are a lot of open sources available. You don't need to worry about commercial tools for that matter. Drug discovery, there are a lot of open sources available and you can definitely employ. And next one is 3D viewers and file formats. Converting one file format to OpenBabel is a well-known free open source software. Converting one form to other format. How do we do? Convert doc file to PDF file. Same way, lot of file formats available. You need to convert from one format to other format. And web servers, like, yes, it goes like this. Molecular modeling. So what are the softwares available? Modeling, like GROMAX is used for simulation, molecular simulation, and Autodoc, and different tools. Whatever you can see, a lot of tools are available. Some are web based, means they have uploaded the web based. You can just upload your protein, upload your ligand, you are going to get the result. Okay, SwissDoc is one such tool. And there are like standalone software like Autodoc and all. You can download, you can perform the analysis. Always the standalone softwares are good. Why? Because you can handle a huge data set. Okay, it is whenever your data set is huge. For example, you want to screen 1 million compounds, because zinc compound or PubChem database might have millions of compounds. Okay, I want to just virtual screen all the compounds, a huge data set. Okay, you cannot handle with web servers because space matters a lot. That time you can download the data sets and you can run the analysis. And this is the protein information, protein RCSP PDB database. You can see Corona 19 resources now. Those who are interested to work on Corona, the nice information structures. Every day they're solving some structures, and there are problems with some structures. They're resolving those problems, and you can identify in the PDB resource. And I was talking about string database, like protein-protein interaction. Okay, I found a target. So you talk about some genetic diseases. Okay, I found this particular drug target. So I will design a drug for this particular target. Next, what happens? Okay, if I inhibit this particular protein, what happens next? Where it goes? Okay, is it blocking any other biological activity? How do I identify? This is string database is one such database, will help you to understand the interactions. Means it will show you what are the interactions between the proteins. It will give you a nice map. I have taken a DDC protein, dopa decarboxyl. You can see here. So this dopa decarboxyl is interacting with these many proteins. If you mutate, any mutation happened in this particular protein, or if you target this particular protein, that might affect other proteins also. So sometimes it might be positive, sometimes it might be negative. So that's what I always tell. Target identification is always a tedious job. And next one is one more database I was talking, like this is EBI, European Bioinformatics Institute. You can also have like how NCBI is having, it is part of European countries. You can have a lot of databases are available. You can go through and identify. And this is the one database I was talking, Therapeutic Drug Target Database. Okay, this is where the database where you can identify the target. Okay, you can whatever the targets information is available, you can take up and work. Because every drug information might not be available. Okay, because people are working and uploading to database. What is the beauty of this database? That's what I say. More than working and publishing papers, if you make some databases, people can utilize those databases. It reduces the burden. If plant compound information is available, if you identify, for example, today I took Tinofora cardifolia, I found out some 1,000 compounds are available, because today analytical technique is advanced. You can identify any compound for that matter. To identify 1,000 compounds are available. What you do, you just publish a paper and leave it. But if you convert those 1,000 compounds into 3D structures and make some database, upload somewhere, definitely people can utilize that and work on that particular model. So databases very, very crucial for bioinformatics analysis. Hope this talk was interesting, and I would like to take any questions are there.
Thank you, TPK sir. Thank you for opening the secrets of bioinformatics. You have completely opened the sources. What are the sources available in online?
Yes.
And better, you can write a book, sir, on this.
Yes, we'll try.
Secrets of bioinformatics. You can write, and we can sell it for huge numbers.
No, sir. We'll make it open access, sir. Let everyone use this. Huh?
Fine, sir. Fine, sir. So people have asked some few questions.
Yes, sir.
So if we want to find a database which can tell us the chemical, which can bind to pesticide chemicals. So they are asking chemical interaction like that. They are asking, is there any databases available?
No, I'm extremely sorry. Till date, I have not come across any such problems. We're basically working on protein drug interaction and all. Chemical interaction as such, I have not come across.
So next question is, which database are reliable for ligands potential target discovery? Is there any, as you have said, no zinc database?
I told, see, PubChem, CHEMBL, they are all authenticated sources. And
Fine,
even
sir, fine.
as I told, ligand information might be available, but the thing is, if you want to go for further analysis, definitely they should look into commercially available. If they have a sophistication, they can purify something and get it, fine. But sometimes we cannot do that particular job. So better they go for zinc databases and all. Maybe FDA approved drugs also easily available in the market. They can consider.
Yes, yes. So how to choose disease modeling target protein from database? For example, how to identify gastric cancer modeling target protein from database? Is there any way to find out the database? I hope it can be done only through literatures, right?
One is literature. Literature is the best source for any disease. What I am telling, the target databases are also developing. They are not like ultimate sources. People are developing. As you are talking about gastric diseases, that might be different route. Gastric cancer may be different causes. It may be due to Helicobacter pylori, or it may be food activities. The sources may be different. What is the cause is again important. What is causing the disease is again important. It's not just you target that particular disease. Cause for the disease is very important. If you analyze the root cause for that particular disease, maybe it will be easy for us to identify the targets.
Fine. So someone asked how one can know that which drug can be used for drug repurposing for other diseases. So this is the hypothesis which we have discussed earlier. And
it is purely like, see, some drugs, some have some anti-inflammatory activity or some activity is known. When they do any particular drug, they know that other activities as drug repurposing, as you are telling, maybe again hypothesis. So for example, HIV drugs today we are repurposing to SARS-CoV. So many other viruses, it might not work efficiently, but we think we are trying. That might work. Even anti-malarial drug we are trying. Because it might target some receptors, some drug targets, it might have a matching. It's purely lock and key mechanism. So sometimes one have a unique lock and key. Sometimes one key can be used for multiple locks also. But in nature, it will not happen like that. According to my knowledge, usually in the nature, it is very unique. Everything is unique. Maybe we need to work on that. Easily, things will not work out.
Fine. And the next one is, in the research papers, 1 million compounds are downloaded from a database. They have given the word, I have downloaded 1 million compounds from the database. Okay, which database should we look into to download? For example, all the compounds that fall into a category of benzimidazole nuclease. Example, they want to filter out. So which database they can work out?
Maybe the person who asked the question should try out this, making a database for that now. So that's what I told. So when you download the papers, even we are working on SARS-CoV-2 and all, when you are working any plant compound, so that particular plant, hundreds of people have done the research and they identified one compound. Maybe we pull all those information. We take from 100 different papers, like good research articles we take, and we try to pull the information. Some of the compounds, in some of the plants, we identified 400, 500 compounds in the literature. Very few numbers, like 100, 200 numbers are only available in the databases. Remaining compounds, we drew it using particular softwares, some softwares. Okay. So again, you have to read.
Cool. Can you explain a bit what is David database? As I heard a lot about this database. Is there any database named David?
No, I have not come across such databases like that.
Fine.
Sorry, no idea.
Normally we identify binding sites from literature. How effectively is identifying the same using softwares which identify the cleft regions?
Yes, good. That's what I think. There are some de novo methods and there are some homology based methods. Like you don't have any similarity, because when you take SARS-CoV example, why I'm taking SARS-CoV, because it's relevant to us now in the present trend. SARS-CoV protease may be similar to other earlier SARS-CoVs, MERS and all. If you are people like they worked from 10-15 years, people are working on MERS, SARS and all, they would have identified the binding site. So when you do homology analysis, you can find out, it is easy for us to analyze the binding sites. Okay, you can rely on such data if it is homology based one. If it is like trial and error methods are there, some what you call docking. There are different docking methodologies are there. That's what it is. Prediction is always prediction. Unless you do some wet lab based analysis, you cannot prove anything, because proving again using in silico tools is not validated. You have to go for
Yes. You have to go for the lab.
Wet lab based.
Wet lab
If
analysis.
if you know bioinformatics means you cannot leave wet lab. That's again you have to go back to wet lab and you have to prove it.
This is a key to find. That's all.
Yes.
Yes, and the last question is any tool to for the preparation of chemical compounds without prior knowledge of programming languages and MySQL, etc. Is there any idea to make a database? They're asking.
Databases, yes, of course, MS Access and all is there. You can MS Access one software. And today, making websites or databases is not so difficult, because there are a lot of tools. Databases will help you. They are kind of how we use MS Office or Windows. That much easy now. Even using Linux has also become easy now, because GUI based, even using Linux is also become easy. Yes, there are even MS Access is one such tool. You can use it for creating simple databases.
Fine. Last but not least, what are the database for the prediction side? Is there any database for finding the prediction side?
What does prediction of binding site prediction?
Yes. Like
maybe
that.
They can go for binding DB and all. That might give you information. Or best way is that if protein is having structure in the PDB database, definitely if they are solved with co-crystallized structure, binding information is available there.
Fine. Thank you.
Okay.
So
thank you,
that's all about the question session. Wait, I'll just go through the YouTube. Any questions have arisen? Nothing. No questions have been arisen in YouTube. Okay, sir. Fine. It's a wonderful and informative message from you and a wonderful presentation. And thank you so much for spending your time, sir. Thank you so much.
Thank you very much for the opportunity. Thank you.